Welcome to Dr. Balaga's podcast for the Curious Doc. Today is June 26th, 2026. This is a video on a Nature Medicine article published 4 days ago on the 22nd of June, open access, titled Biological Aging and Generational Shifts in Early Onset Cancer Risk. Early onset solid cancers are rising in recent birth cohorts. Biological age gap may integrate cumulative exposures and cancer permissive physiology. The study investigators utilized the UK Biobank, 154,169 young adults, an observation analysis of the biobank with partial validation in 10,262 all of the US participants. Systemic aging used PhenoAge, Clemera-Dubol method, and metabolomic clocks was analyzed. Also analyzed was organ-specific aging and plasma proteomics. What did they find? PhenoAge gap rose across cohorts in the UK between 1965 to 1974 versus 1950 to 1954. 23% standard deviation higher. In all of the US, between 1990 and 1999 versus 1965 to 1969, 92% standard deviation higher. In the UK Biobank, for each standard deviation higher of PhenoAge, the age gap predicted early onset solid cancer, hazard ratio 1.08, p-value equals 0.002. For lung cancer, 1.57, p-value less than 0.001. And for gastrointestinal tumors, the hazard ratio is 1.17, p-value equals 0.002. For uterine cancer, 1.31, p-value equals 0.02. In all of the US partially validated the association, hazard ratio was 1.22, p-value equals 0.04. Organ-specific signals linked immune aging to lung cancer hazard ratio of 1.89 and adipose aging to colorectal cancer hazard ratio of 1.60. Limitations of the study: residual confounding is possible. All of the US site-specific validation and UK Biobank organ-specific analyses were sample limited. Generalizability beyond UK and US cohorts and to younger, diverse adults require validation. Key takeaway: greater biological age gap is a research signal, not a diagnostic test. It may help to uncover mechanisms and guide prevention strategies for early onset cancer. Clinical pearls, practical interpretation: accelerated biological aging marked higher early onset solid cancer risk, particularly lung, GI, and uterine cancers. All solid cancer effect was modest but statistically significant hazard ratio of 1.08, p-value equals 0.002. Mechanistic clue: PhenoAge likely integrates inflammatory, metabolic, immune, and tissue microenvironment stress. Organ-specific results point to immune lung axis and adipose colorectal axis. Robustness signal: findings persisted after adjustment for telomere length and genetic predisposition to aging or cancer. Alternative clocks showed consistent direction, particularly for lung and GI cancers. Caution: do not over extrapolate. This observational study does not prove causality, and aging clock assays are not ready to determine individual cancer screening or surveillance. Patient facing implication: message is not you're old, rather cumulative life course exposures may leave measurable biological footprints, making prevention-focused research urgent. Research implication: longitudinal diverse cohorts with repeated systemic and organ-specific aging measures are needed before biological age gap can enter clinical algorithms. This is an infographic on this Nature Medicine article. Hit pause and read at your leisure. Interestingly, there was a Lancet editorial published today titled Cancer in Young Adults, Out of the Shadows? Why it matters: early onset cancer is rising into public view with political attention in the US and G7 countries. Cancer remains age-associated, but adults aged 20 to 49 show increasing incidence in many countries. This Lancet editorial frames this as a health system problem, not an epidemiologic curiosity. What's the epidemiologic signal? Across 42 countries, incidence rose in younger adults for thyroid, breast, colorectal, kidney, endometrial cancers, and leukemia. In 69% of the countries, the average annual percentage change was greater in younger than older adults. Drivers remain unclear. Probably obesity, diet, microbiome shifts, and accelerated biological aging may interact. The clinical challenge: diagnosis is often delayed because persistent symptoms are discounted in younger patients. Screening expansion requires precision. Avoid indiscriminate testing, but identify high-risk adults for early intervention. Young adult cancers may present aggressively, while therapy risks and survivorship needs are undercharacterized. Care model needed. Primary care needs clear referral pathways for unusual persistent symptoms. Care should connect genetics, fertility, preservation, nutrition, and sexual health. Also, psychosocial support and integrative oncology. Fragmented services add burden to both patients and health systems. Research agenda. This editorial says because cases remain uncommon in single centers, collaboration is essential for sufficiently powered datasets. Dedicated young adult cancer programs and early detection interception centers may offer care models. International initiatives such as Prospect are highlighted as critical accelerators. Bottom line, young adult cancer has moved from the margins to global prevention priority. The next step is coordinated multidisciplinary action, identify risk, shorten diagnostic delay, tailor treatment, and protect survivorship. The shadow is lifting. Systems must now learn to see sooner. This is an outstanding editorial. Go ahead and read it. Podcast brought to you by masterbedfacts.com. This podcast is for physicians and healthcare providers. If you enjoyed this video, please like, subscribe, and forward to your friends.